So today our session is going to be probably a little bit longer than usual because in addition to going over the content for the week, we're also just going to go over um, some general information that about getting started in the course. So our objectives for the week are to familiarize students with the main elements in the course and the course syllabus, differentiate between pure and applied science, explain the steps of the scientific process, develop a hypothesis, distinguish between independent and dependent variables, describe the parts of an atom, explain why atoms form chemical bonds, distinguish between ionic and covalent bonds, identify and describe the structure of the four main types of macromolecules. If you have any questions while we're going through it, go ahead and type them into the chat box. So we're going to start with just looking at some of the basics just of the course in general. So I'm going to share my screen with you. So here you should see the course. So we're just going to hit on some of the main parts. So at the beginning of each unit, you'll see unit objectives. And then when you get to the lesson, you'll also see the lesson objectives. So here's the learning objectives for the lesson. Each lesson has somewhere between usually like 7 to 14 or 15 activities. And you have to complete an activity to have the next activity unlocked, so you can't skip forward. That's one question that I've gotten already. As you go through the course, let's let it load. You're going to notice notes in it that it calls them teacher notes, and you'll see a picture of me. And those are all the things that I've added to the course. So the rest of the course, besides the teacher notes, is just what we have from the company. The teacher notes are what I can add to it. So a lot of the times that's where I'm going to put important things. Maybe I've noticed that people are struggling in a certain area. So it's where I can personalize it a little bit more. So here's some teacher notes. We have the syllabus, and then we have both the syllabus and this link here I emailed out to you yesterday, which is the answers to commonly asked questions. A lot of the questions that I get, I've put on there. So that's a good link to refer to and read it if you haven't already. At the beginning of every lesson, there's going to be a what should I know? And these are the notes. So you should be taking notes during the course. This you can either print it or you can save it and type in it. I recommend that you print it out and handwrite on it. I've just found that it's a more effective way of taking notes. People seem to memorize a little bit better when they've actually written it. If you don't have a printer at home, but you think, oh, I can ask my mom to print it at work, or I can go to the library once a week. If you need them sent to you like all at once, like a week at a time or something, you can contact me and that's a possibility. So this is where you'll take notes. You absolutely should be taking notes. Keep going. So here you'll see some things. Here there's a Bitmoji, but normally it's a picture of a cat reading a book. And then there's this box that doesn't look like anything. This is a recording of the text. So wherever there's large pieces of text, like we're going to see one under here, this little box is of a teacher. So it's not a robot voice. It's actually one of the teachers here reading all of this text. So you can download them to your phone or your iPod, and you can listen to them at other times, or you can just click on them here and listen to them. But in most of them, there's a cat reading a book, and that's how you know that's going to be the reading. So here's like the text for the lesson. This is what you're taking notes off of. So there's cat reading a book, and then that gray square is our recording of the reading. There's videos in most of the courses. In every lesson, there's a discussion board. Discussions are 15% of your overall grade. And you'll see for You can't see it on this one, but if I was in one of your courses, there's usually like a colored box right at the top that's it's a pin of what I wrote. Some of those I've just broken the question down into like simpler parts. Some of the discussion boards have just been kind of notorious for nobody gets them correct, so I've completely changed the question. So whenever there's a pin on every single discussion, sometimes it'll just be breaking down the regular one, or sometimes it'll be a completely different question. Please do the pinned one. That's the one that I prefer. Then you'll always see a Bitmoji, and that Bitmoji is where I've linked the vocabulary terms on Quizlet. This lesson only has 3 vocabulary terms, so it's not going to be very much, but some of the lessons have a lot of vocabulary. So if you're not familiar with Quizlet, you can kind of explore that. You can do it just like flashcards. My favorite one is to do the match. It's kind of like a game. So you start and it'll time you and it'll put all the vocab words. Like I said, this time there was only 3. And you have to you have to match them together. So if you match them incorrectly, then they turn red and they just bounce back. If you match them correctly, then they go away and it'll time you so you can try for a better time. Your checkpoints, there's checkpoints in every lesson and it's kind of like a quiz. You get 3 attempts. So I always suggest that if you get lower than an 80%, if you still have remaining attempts, to go ahead and use them. You want to get as high a score as you can, and also it'll take your highest score. So it's not like if you got an 80% and the next time you got a 70, it takes the last one. It takes the highest score. So there's nothing to lose. Over on this side here, there is a little envelope, and that's where you can message me directly from your course. On the checkpoints and exams, what I recommend is that you open 2 tabs, one where you're taking the assessment and the other one where you can go through the course so you can look things up. But also if you've taken notes, that's a great thing that you can use on your tests. So, all right, so that's just a quick look at the course. Let's go back and we're going to go through the syllabus. Hopefully you've already read through the syllabus. I emailed it yesterday, I believe. So this is Biology A. There's my contact information. There's quite a few different ways that you can contact me. You can message me. You can IM me. You can call me. This is the number that goes directly to my desk, so it doesn't go to a receptionist. It just goes straight to me. Or you can email me, and there's my email. Here's just a brief description of the course, so what you should be learning overall in this course. Here's the objectives. The standards, we have both the Primavera curriculum map that kind of shows you what you'll be learning and in what order, and then we have the Arizona Department of Education Science Standards. You can take a look at both of those. For this class, we have 4 projects. It used to be 10, so it's gotten much more reasonable. For the first project, which is in lessons 2 through 4, I feel like it's 2 through 5, but maybe not. You're going to need cups or glasses. You're going to need clean measuring spoons, stirs, whole milk, buttermilk, masking tape, and a pen to label it. For the second project, which is in Unit 2, you're going to need a potato, food coloring, cups, and then you'll need like a knife to cut the potato. In the next project, which is in Unit 4, you don't need any materials. This is going to be a closed reading. It's a CER, which stands for Claims, Evidence, and Reasoning. And then the last project is in Unit 6, and that one is a research project. So already in the syllabus, what materials you'll need. It's always best if you can do the lab yourself, if you can do it at home. But if you can't, you'll see in the lesson as you go through that it'll say like no materials, no problem, and there's a slideshow that you can watch to get the data from. Even if you watch the video as opposed to doing the actual activity, you'll always still fill out the same paperwork for it though, and we'll get to that. Here's the coursework, the different ways that you can earn credit. Here's how that's broken down. So you can see the synchronous sessions are 15% of your grade. Exams, this is unit exams, are 10%. Checkpoints, which you can do up to 3 times, are 10%. Discussions are 15%. And for this course, the discussions are designed to be research questions mostly, so those are going to require a little bit of time. Projects are 30%, workbooks are 5%, and the final exam is 15%. So you can see projects are a huge chunk of your grade, so make sure you're really putting a lot of effort into those. If you come into the course and you just do what's built into the course and you don't do that extra stuff where you have to go outside, which is synchronous sessions, projects, and discussions, people think that they can get through the course with that. And even if you get 100% on everything, if you don't do those categories where you have to do something like outside work, the highest grade you can earn is a 40%. So you can't even pass. So make sure that you're really putting effort into those. And then, of course, if you ever have any questions, I'm here, ask. There's some things that are automatically graded. That's the stuff that's directly in the course. So the workbooks, the checkpoints, and the exams. And then projects and discussions and synchronous sessions are graded by me. So once you submit, they won't automatically have a grade. You'll have to wait until they get graded. So it's usually just a couple of days that you have to wait. Here's the synchronous sessions, which is at what you're at. The day and time for the session is always going to be Tuesdays at 1 o'clock. If you can't make it after the live session, I'll take the recording and I'll put that in the announcements along with an exit ticket, which is like a little quiz on the session. So you can do that to still earn your points. Expectations. It's pretty much standard for just being in school. We expect that you do your work and that your work is actually yours. So be really careful with using work that somebody else wrote. If you get it from a source, make sure you cite it. And then, of course, never use another student's work as your own. And then there's a link here to the parent-student handbook, which goes over student conduct. These are rubrics, so how each of these things is graded. We have the 4 different projects, and then synchronous and asynchronous sessions and the discussions. The pacing calendar. This is going to tell you what lesson that you should be on each day. This one is for the entire block. And then if you looked in the course announcements, I have it per week that I put in the course announcements. So it's 1 lesson per day for each weekday, or 5 lessons per week. And then here's some additional support. Thinking Storm is an outside service that we use. It's free, but it's tutoring. Student support and guidance, if you have any questions about enrolling in courses or where you are in your graduation track. Tech support, if you're having problems with a technical aspect of the course. About 90% of the time when we get technical problems, it's that a student needs to clear their cache and cookies on their computer. But if you've done that and still you're having trouble, call that tech support. And then the Learning Lab, finally, is for students with an IEP or a 504. If you don't have an IEP or a 504 and you need tutoring outside of the hours when I'm here, you'll use the Thinking Storm. Okay, so that's kind of the gist of the general stuff of the course. Does anybody have questions on that? So if there's anything that was confusing, please go ahead and ask. I know I went through that fast. No? Thank you for replying, Genevieve. Does anybody else have questions? Okay. All right. And I'm here in the office every weekday. So if you ever need help with something, please don't hesitate to contact me. Okay, so now we're going to go ahead and we're going to look at the content for this first lesson. We're not going to go through every single thing, just kind of hit the high points, what I've noticed that other people seem to struggle with more often. And probably if you've taken a science class before, some of the stuff in this first unit looked a little bit familiar. It won't all look familiar. So the first topic they talk about is pure versus applied science. So pure science is just learning for the sake of learning. So researchers want to understand how and why the world works. So it deals with knowledge discovery. You're learning something new. And then applied science, researchers aim to solve problems with that information from pure science. So it deals with knowledge use. So when you're just learning something, applied, you're applying what you learned. So an example is in this first picture, pure science, a scientist just discovered a new chemical. They published it, they told others about it, it's a new chemical. And then applied science is maybe another scientist takes that new chemical and they develop it into a cosmetic line. All right, so I'm going to give you a link, and there's a lot of you in here, so we're not gonna, I don't know if I have enough for everybody, but go ahead and log into this link. And you should all see a lino. So I have pictures of scientists doing something, and I want you to, I'm going to assign you a picture, and you're going to tell me if that picture is pure science or applied science. And how you answer it is you're going to take a sticky. I like the little clear ones, oops, the best. So you're going to click on that, and it's going to make a box. You can change your color if you want, and then you're going to type your answer. So you're going to type pure science or applied science. And then you'll click post, and then it'll have your answer. And then once you have your answer, then you're going to grab it and you're going to drag it to whichever one you were assigned. So you won't write answer, of course, you'll write pure science or applied science. Okay, so I'm going to go ahead and I'm going to assign you a number. If you're wrong, it's okay. This is just some practice. So Aiden, you're going to do number 1. Ansley, number 2. Genevieve, number 3. Griffin, number 4. Christina, number 5. Mallory, number 6. Denise, number 7. Sebastian, number 8. Tiffany, number 9. And Vanessa, number 10. So you'll get a sticky. For the number that you did, you'll write if that looks like pure science or if it looks like applied science. You just need an answer. Yeah. If you feel like it needs explaining, you can put that on there, but you can just write pure science or applied science, and then you'll put the sticky next to the one that was assigned to you. So I'll give you a few minutes to get that done. So if it looks like they're doing science just for the sake of learning something, or if it looks like they're using science to develop something new. So pure science is knowledge discovery. Applied science is knowledge use. So you're taking the knowledge and you're applying it to a new field. Okay, so I'll give about 1 more minute. If you realize that you messed up and you needed to change it, you can click on it and you should be able to change it. Or if you click the little check mark, it'll say PO off and you can remove it and you can put a new answer on there. Okay, let's go ahead and go through our answers. Number, oops, number 1, so we can see some people working in a lab. Looks like they're working with some glassware. So we said this one is applied. Why do we think that one is applied? And some of these can have more than one correct answer, but you just might have to explain why you thought that. So, okay, so a bunch of you were typing and then nobody typed anything. Okay, good. I would agree. And like I said, some of them have, you know, if you can explain it, then maybe it's different. That to me looks like pure science. You can't see that they're applying it to anything new. It just kind of looks like they're um, like students, like they're learning for the sake of learning. Okay. You can see a couple of you are typing, so I'll wait a minute. All right, next, number 2. You can see this guy, he's got some glassware, he's wearing some goggles. What do we think about that one? Okay. So like I said, some of these there can be, it's more about, I guess, kind of the explanation. If you were to see this on like an exam, I would say pure science because it doesn't look like they're applying it to something else. But yeah, if he had pure science because he's testing something, okay. Yeah, so it's a little bit subjective. Um, and that you were able to explain it, Ansley, makes like the difference. But if you were to see this on an exam because you can't see that he's developing something new, I would say that that's pure science. Number 3, we can see this guy with this, I don't know, maybe motor, an engine or something. So he said that's applied science. So does that seem just like he's learning something new, or does it look like he's applying it to a new situation? So there's a few of you typing, so I'll wait a minute. All right, so it looks applied because it seems like he's applying it to something new. Looks like he's applying it to a new situation. He's building a model. Good. Looks like he's taking knowledge of engineering and applying it to create something new. Perfect. These are all great answers. That was absolutely looks like applied science to me. Number 4, this person just has some test tubes and is looking at some stuff in a science setting. So I would agree that looks like pure science. Number 5, again, in a lab setting, looking at something through a microscope. That also seems like pure science. Number 6, these guys just using some machinery in a lab. So pure science, good. And you'll notice kind of a trend that when they're in a lab, a lot of the times they're just learning for the sake of learning. When you're kind of out in the field is when you're applying it to something new. So number 7, we have a drone here that looks like a dragonfly. So absolutely, they took the science of, the engineering and also I'm sure a lot of the biology that they learned about a dragonfly and were able to create this drone. So that's applied. Very good. Number 8, used science to create these weapons for the military. So that's applied. Number 9, used what they learned about anatomy and physiology and some engineering to create this device for a man to walk who could not walk. So that's applied science. And number 10, so they're in some type of a manufacturing setting. And this one is always the one that I feel like can go either way because it looks like they're creating something on a large scale. Maybe it could be applied, or maybe they're just working on discovering something new. So there's a couple of them that I feel like they could be either depending on your perception of it. So, all right, good job. So let's go back to the PowerPoint. Okay, so that was pure and applied science. Next, the scientific process. Our first step is to make an observation or ask a question. Then after you've made your observation, you have to form a hypothesis. Then you conduct your experiment, analyze the results, draw conclusions, and then after you've repeated this and you know that your work is valid, then our last step is always to communicate the results or to publish. And you'll be using the scientific method in the yogurt lab, which is in Unit 1. Hypotheses. So we'll be writing many hypotheses over the length of this course and your other courses. And this always seems to be a little bit tricky. It's maybe a different format than you're used to. So the format that we use here is if A, and A is an explanation for the effects of the independent variable, then B, and for B you choose either increasing or decreasing, the independent variable will C, increase or decrease the dependent variable. And I know that sounds confusing. So we're going to do a couple of examples. Okay, so for our first question, we'll say, how does the size of a dog affect how much food it eats? Okay, so there's our question. So what are our two variables? First of all, what is our independent variable? So the independent variable is what is it that we, the scientist, are altering? Are we altering Okay. So we're going to take a dog and we're going to give it different amounts of food? Or are we going to take different size dogs? Okay, so the size of the dog affect how much food it eats. Do you think we'd change the size of the dog? Or do you think we'd change how much food we give it? I know it sounds tricky. Okay, good. It is going to be the size of the dog is what we're going to change. So we'd want to do a big dog, we'd want to do a little dog, we'd want to do an in-betweeny size dog, and then we'd see how much it eats. So it'd be if, and we state our relationship. So if the size of a dog affects how much food it eats, so that's A, then we do our then, and then we say increase or decreasing. So that you can just pick. So do we want to say increasing or decreasing? So I'll go with the first one, decreasing. Okay, then decreasing the size of the dog. So that's the B part. And then C will, if we decrease the size of the dog, do you think that will increase how much it eats or decrease how much it eats? Decrease, good. Will decrease the amount of food eaten. So does that kind of make sense? So we have if we state the relationship, then increase or decreasing the independent variable will increase or decrease the dependent variable. So we're going to do one more, and I'm going to pause for a little bit because I want you to try to come up with the answer yourself, and then we'll go over it. So our question is going to be, does fertilizer make a plant grow bigger? Okay, so I want you to use that question and try to formulate a hypothesis using the format that we said. So I'll give you a couple minutes. Okay, so I'm seeing some good answers. The most important part is that we use the format. So our first line needs to be stating the relationship. So if what we're comparing the relationship is the fertilizer and the height of the plant. So if fertilizer affects the height of a plant, or if fertilizer affects the size of the plant. So first, we're just stating a relationship. We're not stating how it affects it. So if fertilizer affects the size of a plant or the growth of a plant, then, and then the second part we all seem to get correctly, which then increasing or decreasing the amount of fertilizer will increase or decrease the size of the plant. So that last part is where your opinion, your prediction comes in. Whereas the first part, we're just establishing what two things are we testing. Okay, so if fertilizer affects the growth of a plant, then it looks like most of you said increasing. Increasing the amount of fertilizer and then looks like most of you thought that would increase the size of the plant. Okay, so you guys seem to do pretty good at that. Does everybody feel a little bit more comfortable with that format? Because that's usually the biggest mistake that we see in the labs is formatting the hypothesis correctly. Good. Okay. And if you ever need help writing a hypothesis for any of the labs, just contact me and we can work on that. Or we also have our tutoring time, which is Thursdays at 3 o'clock, that you can log in and just get help with something and leave. You don't have to stay for the whole time. And then if you ever need help that's outside of that, of course, you just text me or email me or call me. All right, moving on. You guys are doing great. Thank you for participating. Okay, so we said our hypothesis, our variables, we talked about this some already. The independent variable is the one that is purposely changed or manipulated. That's the one that you, the scientist, are altering. And then the dependent variable is the one that's observed or measured, so it's the responding variable. So if we were to think of it in a sentence, the independent variable influences the dependent variable. Dependent variable changes because of the independent variable. So you will have to identify these variables in your labs. And then the control, when we refer to the control, that's the one that's under normal conditions, so we use it as a source of comparison. So like with our plants, if we were comparing the fertilizer, we would need one that we don't give fertilizer to, so we can see if the fertilizer was actually doing anything or if that's just what the plant did naturally. So our control is their normal conditions that we can use for comparison. Okay, atoms. An atom is the smallest unit of matter. It's not the smallest unit of life. We get that confused sometimes. The smallest unit of matter. And it has 3 subatomic particles, so smaller than an atom. And those are protons, neutrons, and electrons. So first, we have our protons. And those have a positive charge. And they are located inside the nucleus. Next, we have neutrons. They have no charge. They're neutral. And they are also located in the nucleus. And then finally, we have electrons. They have a negative charge. and they are located in orbitals or just areas outside of the nucleus. So in our picture here, because we have protons are positive and electrons are negative, those cancel each other out. An atom means that it's neutral. So there's equal amounts of protons and electrons. So if I know these are my electrons because they're outside the nucleus, and there's 3 of them, then the red ones have to be my protons because atoms have equal numbers of protons and electrons, which means the blue ones are my neutrons. Any questions on that? All right. Next, bonding. We have 2 main types of chemical bonds. We have a covalent bond and an ionic bond. Covalent is this first picture here that we can see at the top, the atom on the left has 1 electron, and the atom on the right has 1 electron. So they share, and so these atoms, the electrons, will go around both of them. And the electrons will spend equal time around both of them. So that's a covalent bond. So think if you are a co-captain, you would share the responsibilities of captaining. So those, they share them. In an ionic bond, one atom is going to give away an electron, and the other atom is going to accept that electron. So we know the proton is positive, the electron is negative. So if this one on the left gave away an electron, now he has 1 positive and no negatives. So the overall charge is positive, so we call it a positive ion. It gives this electron away, so the one on the left then will have 2 electrons, 2 negatives, and 1 positive. So it has an overall negative charge, so we call it a negative ion. A cation and an anion, yep. And then just like magnets, we have a positive and a negative, and they attract each other. So that's how an ionic bond works. And so we call it an ionic bond because when they don't have equal protons and electrons, then they're called an ion. So we had a positive ion and a negative ion, and then opposites attract. Okay, then next with chemistry, we don't do tons of chemistry in this class, but the chemistry we do talk about is macromolecules because these are the ones that are important in life in our bodies. There are 4 macromolecules. They are carbohydrates, lipids, which are fats, proteins, and nucleic acids. And because macro, they're big molecules, they're made of smaller molecules put together. So the subunits are the smaller molecules that we put together. To make carbohydrates, we put together monosaccharides, single sugars. And then what we end up with is a polysaccharide, many sugars, which is a carbohydrate. And some examples of carbohydrates are starch, amylose, and cellulose. Cellulose is what we find in the cell walls of plants. It's what makes them crunchy. Next macromolecule is lipids, and the smaller molecules that they're made of is a glycerol and fatty acids, and this is just in general. Some examples of lipids are fats, phospholipids, steroids, and waxes. Next macromolecule is a protein, and those are made of amino acids. And then what's in parentheses there is what makes an amino acid. An amino acid is made of a carbon, an amino group, a carboxyl group, and a hydrogen with an R group, and we'll see that in the next picture. And some examples of proteins are actin, elastin, keratin, and membrane proteins. And then the last macromolecule is nucleic acids. And the smaller molecules that nucleic acids are made of are called nucleotides. And then the N in nucleic and A in acid are the NA in DNA and RNA. And we'll learn more about those in a later unit. So now we're just kind of quickly presenting them, and then we'll learn more about them in other units. So here's some pictures. So we said carbohydrates are made of monosaccharides. This glucose is a picture of a monosaccharide. So we put together lots and lots and lots of those, and we get a carbohydrate. And here's one carbohydrate, which is amylose, and we can see it's just glucose, 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 and then it just keeps going. Then up in the middle is a lipid, and this specific lipid is called our phospholipid bilayer, which is what our cell membranes are made of. And it includes it's called a phospholipid because it has a phosphate group, and then it has fatty acid tails. So we can see it kind of looks like a head and a tail. So we have the phosphate, and then we have the fatty acids. Then our protein, we said the structure those made of is called an amino acid. Here's the structure of an amino acid. All amino acids are going to be exactly the same except for the R group. So they're all going to have an amino group. They're all going to have a hydrogen. They're all going to have this central carbon. They all have a carboxyl group. And then the R group changes. That's what makes one amino acid different from another one. And then we hook those all together, a chain of amino acids, to make a protein. And then lastly, we said a nucleotide is the individual subunit in a nucleic acid. So what's in this box here is a nucleotide. It has a phosphate group, which is the little black circle. It has a 5-carbon sugar. We can see 1, 2, 3, 4, 5. And it has a nitrogen base, which is either the C, the T, the G, or the A. And then we hook those together to get a nucleic acid. And this one here is DNA, but we also have RNA. We'll learn about those later. So this is just the basics of what makes each of the macromolecules. And that's the end. So, does anybody have any questions? It can be about anything. It can be about the information in the first unit or about something general to the class. No? Okay, I do like to hear from you as often as I can. So through the IM or through the messaging or email, make sure, keep me in the loop. Let me know how you're doing. Let me know if you fall behind, if there's anything that I can do to help you. But I'm here all the time. I'm not just a computer. I'm a real person. So you can get help whenever you need it. All right? So thank you for coming. If you have questions or need help, probably especially with the lab or exam, Thursday at 3 o'clock, we'll have just like open tutoring that you can stop by. And if you have questions, stick around. If you do not, you are free to go. Thanks for coming. Thank you. Bye-bye. Thank you.